Cardiovascular Research Discoveries seminar series, which is co-organized by Council for Basic Cardiovascular Science of the European Society of Cardiology, together with Cardiovascular Research, the Basic and Translational Science Journal of the ESC as well. As you remember, our seminar series is organized in a way that we invite an established scientist, and today we are happy to host Professor Stanley Nattel and up-and-coming researcher who has already hugely contributed to the field. And today we have an honor of Dr. Svetlana Reilly joining us to talk and discuss between themselves and with us the new targets in atrial fibrillation. The first talk will be delivered by Professor Stan Nattel, whom probably you all know very well, but I will introduce him anyway, as he is a professor of cardiology and internal medicine, and in particular electrophysiology at Montreal Heart Institute at the University of Montreal. He's known, a well-known authority in atrial fibrillation, an associate editor of Cardiovascular Research for many years now, and an editor-in-chief of Canadian Journal of Cardiology. So welcome, Stan, and as promised, here is Stan. And please remember to ask questions through our question and answer part. The discussion will take place at the end of our seminar, but please ask questions throughout the talks. Thank you.
Thank you very much for the opportunity to speak, Tom. I think you have to, someone has to stop your
yeah,
sharing.
I think it's ready.
Okay, there we go. And let's hope this will now work. You let me know if you see my screen. You see my talk?
Yeah.
It's occupying full screen? Yes?
Yes.
Okay. And do you see the pointer?
Yeah.
All right. Wonderful. Okay. So I'm going to begin with a few words on the AF therapeutic environment landscape. This is from a paper that we wrote in a series of articles in a spotlight issue of Cardiovascular Research on translational research in AF. So there are a wide range of factors that motivate therapeutic discovery in the range of AF. The first is that it's an extremely common clinical problem. Anyone who's a clinician and spends time in the emergency room or on the cardiac wards has seen many, many patients with AF, being readmitted with AF, having complications of AF. And unfortunately, doctors in neurology have seen too many patients with strokes from AF. The condition has high morbidity and is associated with an increase in mortality. And of course, the complication rates in terms of ventricular function and stroke and possibly even dementia currently under investigation are very significant. And finally, the major associated health costs and economic burden provide a financial incentive for advancing discovery in this area. So it's certainly an area that's ripe for advancement. However, as we all know, there are many challenges to development. The first is the complexity of the clinical problem. There are many different types of patients, many different types of AF, and so any attempts to treat all patients as one or to use a convenient screening group for a drug may fail because of the complexity of the arrhythmia. Then, of course, there are difficulties extrapolating from animal models to humans. And in addition, specific limitations of individual species and experimental paradigms. Each of the available species has their own intrinsic advantages and often major disadvantages, and all of those need to be considered when attempting to develop new therapeutic approaches. There are also regulatory considerations, which are complex and which in many cases require large studies to ensure that there's no increased risk of proarrhythmia or death. And finally, the cost of development in AF, as for most groups of drugs today, is very considerable. So thus, while there's promise in this field, any new therapy for development must have the following properties. Obviously, it's got to be effective or it's not worthwhile. And in particular, it has to be relatively non-toxic, because many patients with AF or at risk of AF are not terribly ill, and any drug that's significantly toxic will not be able to be used in a reasonable proportion of the overall population. So overall research to identify safe and effective new targets is clearly key. And here I'll focus on one such potential target, cellular inflammation, which has emerged over the past few years as a very interesting target that might have potentially relatively non-toxic approaches. So this is an outline of my talk. I'll talk about evidence for the role of inflammation of AF, recent experimental insights, including evidence for cardiomyocyte inflammatory signaling and mechanisms of AF and right heart disease as a paradigm in which the role of inflammation resolution can be studied. So this is really, to my knowledge, the first study that proposed that inflammation might play a significant role in AF. It's from Mina Chung's group. David Van Wagner was a prominent co-investigator with Mina on this study. They looked at plasma CRP levels and correlated them with the presence of AF, and noted that in large numbers of patients from the Cleveland Clinic, patients with AF, whether lone or associated with structural heart disease, had a higher blood level of CRP. And there was a graded relationship between the type of atrial fibrillation, paroxysmal, having a lower CRP level than persistent, and the presence of an increase in CRP. This slide shows the classical view of the role of inflammation, where macrophages and other leukocytes play a central role initiating inflammation and associated changes via release of cardiokines, angiocrines, and all kinds of other cytokines. Cardiomyocytes and fibroblasts, from this point of view, while central in the pathophysiology of AF, act as passive mediators. However, recently, evidence has been accumulating that cardiomyocytes and even in fibroblasts, which I won't speak about today, can show inflammatory signaling within themselves, that they have all of the messaging, all of the machinery necessary to induce cellular inflammation. So this slide is complicated, but I'll walk you through it. It's a necessary review of the components of inflammatory signaling. So some of the key elements of the inflammatory system include procaspase 1, ASC, which is apoptosis-associated speckled protein containing a caspase domain, and NLRP3, which has a very long name that keeps changing, and I'm not going to try to read it, but you can all see it on the slide. When there are increases in the intracellular concentrations of these compounds, that's called priming, and it prepares the cell for inflammatory activation. So that priming is the first step in activation. The second step is activation itself, which involves assembly of the NLRP3 components into an inflammasome. You see here, this little orange bit is the NLRP3, the blue ball is ASC, the green is procaspase 1. All of these come together and activate. They then convert pro-interleukins, such as pro-interleukin 1 beta and interleukin 18, into their active byproducts, interleukin 1 beta and interleukin 18, and cause these cytokines, along with others, cause the inflammation. And as I mentioned before, normally it's understood that these chemicals play a major role in inflammatory and immune cells like macrophages, but we don't think of cardiomyocytes, fibroblasts, and other cell types as having systems that mediate this. Dobromir Dobrev and Na Li, from in collaboration with Xander Werens, looked at the evidence for the presence of NLRP3 proteins in atrial cardiomyocytes, both from AF patients and from dog AF models. And what they noted was an increase in caspase P1, P20, which is an indication of activation by breakdown of caspase 1 into caspase P1, P20 in paroxysmal AF patients, shown here, and evidence of priming in chronic or long-term persistent AF patients with increases in both NLRP3 and ASC, as well as caspase P1, P20. And in the dog model, we also noted an increase in all of these components. So there's evidence of increases in cardiomyocyte NLP3 inflammasome priming and triggering in AF. Na and Zander then went on to create a mouse model with constitutive cardiomyocyte restricted NLRP3 activation. And they noted that mice with cardiomyocyte restricted NLRP3 activation developed spontaneous arrhythmia with large numbers of premature atrial complexes, as well as inducible AF. And this is an example in a control mouse with no inducible AF, and in a knock-in mouse with inducible AF, and a clear and substantial increase in the quantity of AF that could be induced, the percentage of the time in which AF could be induced. Interestingly, that was suppressed by administering an organic NLRP3 blocker. So cardiomyocyte overexpression of activated NLRP3 produces an AF promoting substrate. It also produces structural remodeling. These are images of hearts from a control and a cardiomyocyte knock-in NLRP3 activated mouse. These at 3 months, these are images of Masson trichrome stains where fibrosis is shown as blue. And you can see that there's increase in atrial size, an increase in the activation of MEF2C, which is an important transcription factor, and a decrease in ARCAN 1.4, which is an anti-inflammatory compound. Fibrosis is substantially enhanced, and there's increased production of collagen 1 alpha, as well as galactin 3, a well-known fibrotic mediator and biomarker. So cardiomyocyte NLRP3 overactivation causes atrial structural remodeling with prominent fibrosis. I don't have time to go through all of the original data here, but it also causes additional relevant profibrillatory phenotypic changes, including abnormal calcium handling, DADs, and atrial ectopic activity, as well as ion current remodeling with increased potassium current that causes reduced action potential duration and refractoriness. So basically the perfect storm for AF generation. Na and Zander then went on to look at the effect of NLRP3 knockdown, or knockout in fact, on spontaneous AF in an independent mouse model, which is cardiomyocyte-directed CREM mice. And so they compared CREM transgenic mice, a well-established model of spontaneous AF in the mouse, with a CREM transgenic crossed with an NLRP3 knockout mouse. And these are the data for procaspase 1. You see it's reduced. Procaspase P120 is reduced with a double knockout. The CREM transgenic had atrial ectopy, suppressed in the double transgenic. And then if we look at the overall levels here, you can see an increase in procaspase 1 and P120, an increase in spontaneous AF, an increase in left atrial area, all of which were suppressed in the double transgenic mouse. So cardiomyocyte NLRP3 inflammasome overactivity happens in models of spontaneous AF, as well, of course, as in human and dog models of AF. It's sufficient to cause AF in mice and is an essential mediator of AF promotion in an independent model, the CREM mouse. Cardiomyocyte NLRP3 inflammasome activation appears to be a causative component of the pathophysiology of AF in persistent and chronic AF in man. What about other forms of clinical AF? So this is work from Jordi Hyman and Dobromir Dobrev in collaboration with Nan Zander. They looked at NLRP3 inflammasome levels in components in patients who had postoperative AF. So the paradigm for this study was atrial tissue samples were taken, cardiomyocytes were isolated and studied, and then patients were observed to see whether they developed AF or not. And the question was, what is the pathophysiology, the cellular pathophysiology of individuals who will subsequently go on to develop AF in the next few days after surgery? So you can see there was no significant difference in NLRP3 expression, but a significant increase in ASC, pro-caspase-1, gasdermin, the full-length gasdermin, and pro-interleukin-1 beta. So all of these components reflect increased priming in patients who went on to develop postoperative AF, and an increase in caspase-1 inflammatory products and components of gasdermin. Gasdermin D is an ion channel that is opened up in the membrane by inflammation, and these are the breakdown products that form the channel or that indicate formation of the channel, and you can see they're significantly increased in patients who go on to develop postoperative AF. There's actually a lower level of interleukin-1 beta, which they believe is due to an increase in permeability to the cell to interleukin-1 beta that then leaks out of the cell. So you can see increases in priming, increases in activation, and what are the consequences of that? They studied the effect. So these cells are ready to develop postoperative AF, but the individuals don't have AF at the moment of surgery. So obviously something more is needed, and we believe that the something more is inflammation. So Dobr's group infused interleukin-1 beta to mimic cellular inflammation, to control cells and to cells from patients with postoperative AF. And you can see that in the control cell, there were very few spontaneous calcium releases, whereas in the AF patients, there were quite dramatic calcium releases. The number of spontaneous calcium releases and their incidence was greatly increased in patients who developed post-op AF. So this creates a scenario or a paradigm in which cardiomyocytes of patients who develop different forms of AF have activated NLRP prime and activating NLRP3 inflammasomes, whether other cellular components in the heart also participate, such as leukocytes, adipose cells, and fibroblasts. We don't really know yet, but work is ongoing on that. This activates CAM kinase 2 and phosphorylase ryanodine receptor, resulting through a variety of mechanisms in AF promotion. And depending on substrate and regulatory factors, that can produce clinically manifest paroxysmal post-op or persistent AF. So now I'm going to spend a few minutes talking about mechanisms of AF and right heart disease and some recent interesting work on inflammation resolution. This work was done by Roddy Hiram in my lab. I apologize, I haven't updated the citation. It's been published in JACC. So Roddy created a rat model using monocrotaline injection to cause pulmonary arterial disease and hypertension. Rats were injected with monocrotaline at day 0 and studied 21 days later with echocardiography, hemodynamics, in vivo electrophysiology, optical mapping, and biochemistry. And paroxysmal arterial hypertension rats were compared to rats with an AF substrate caused by acute anterior MI. So this was a form of AF, if in fact it happened, which it did, that was due to right heart disease in the pulmonary artery hypertension rats, whereas those with acute MI had a form of AF due to left atrial disease. In a follow-up study, novel pharmacological approach was used, which I'll talk about shortly. So these are the original results from the pulmonary hypertension rats. This is in vivo electrophysiology testing through transesophageal stimulation. You can see The incidence of induced atrial arrhythmias went close to 100%. P-wave duration increased slightly, reflecting remodeling. These are examples of in situ electrophysiology testing, and the results here show, again, an increase in induced AF, increase in AF duration. So monocrotaline-treated rats have increased susceptibility and sustainability of AF. This is analogous to the many patients who develop AF in relationship to chronic obstructive lung disease and to congenital heart disease with pulmonary hypertension, as well as other conditions. These rats showed prominent structural remodeling with fibrosis in the right atrium. This is a monocrotaline treated compared to a control rat. The blue is fibrous tissue and an increase in right atrial dimension. Interestingly, there was also left atrial fibrosis, so presumably systemic activation of profibrotic factors, but no change in left atrial dimension because no hemodynamic load on the left atrium. So monocrotaline causes marked right atrial fibrosis and dilation, moderate LA fibrosis, but no dilation. And looking at these rats, we noted evidence of an increase, but not statistically significant, in NLRP3 in the right atrium of the monocrotaline treated rats, and statistically significant increases in caspase 1 and caspase 8. Large increases in interleukin-1 beta, but a lot of variability, so not statistically significant. So inflammatory signaling gene expression is enhanced in monocrotaline atria, and you'll notice here we also have data for the left atrium and MI, and we also noted changes in that as well. So we decided to try a drug intervention to try to prevent the development of this substrate. And there's an interesting theory which has developed in the last 10 or 15 years, which holds that long chain polyunsaturated fatty acids have products that promote the resolution of inflammation. And so rather than thinking of inflammation as something that happens or doesn't happen, there's this notion that inflammation normally is triggered by a systemic stressor, and then there's a process of resolution which causes the inflammation to resolve. When it doesn't resolve properly, it causes chronic disease, but there are products of these polyunsaturated fatty acids that can promote resolution of inflammation. And one of these is the resolvin D class, which we decided to use in this study. So we took rats who were treated with monocrotaline and gave them, this, by the way, is published in Cardiovascular Research. It just came out in the same AF issue that I presented the overview on translation before. So in control, there was no AF induced. With monocrotaline, there was a substantial increase in AF inducibility, which was reduced substantially with resolvin D1. So these rats got monocrotaline. The groups at the right all got monocrotaline 21 days before their final study to induce pulmonary hypertension, but one group got resolvin D1 to promote resolution of inflammation and the other did not. The degree of fibrosis was significantly attenuated by resolvin, and the right atrial dimension was not changed. Interestingly, the hemodynamic load was also not changed, so the pulmonary hypertension was not substantially affected. And in the left atrium, again, the degree of fibrosis was reduced. So resolvin D1 attenuates in vivo AF vulnerability and atrial fibrotic remodeling in monocrotaline rats. Looking at the effects on inflammatory mediators, this is from a non-directed gene microarray analysis. You can see dramatic increases. These are fold increases, so 32-fold, 23-fold, 18 and 15 in the expression of interleukin-1/6, CCL2, CXCL2, and CXCL1, which are all cytokines and inflammatory mediators. These were substantially reduced, 29-fold and 31-fold by co-administration with resolvin D1. This is on the left side. This is on the right side, and again, you see similar changes. So resolvin D1 strongly attenuates changes in atrial inflammatory cytokine and chemokine genes induced by right heart disease. So in conclusion, atrial cardiomyocyte inflammatory signaling involving NLRP3 activation appears to be a common signaling system implicated in various forms of AF in man, paroxysmal, persistent, and postoperative AF. All show activation of the NLRP3 system. Downstream pathways include calcium mishandling, structural remodeling, and possibly ion channel expression changes. Molecules that promote the resolution of inflammation may be useful in preventing pro-inflammatory remodeling, and inflammatory signaling in AF is poorly understood and may present interesting targets for innovative therapeutics. I want to conclude by pointing out that when looking for new ideas, I'm painfully aware after many years in research that many don't work, and of those that do, many turn out not to be practical. But remember, it takes only one idea that succeeds to revolutionize the area. So keep that in mind when you're struggling in research. Thank you very much.
Thank you very much, Stan, for extremely interesting scientific data and also some words of wisdom. And when introducing the seminar, I did not emphasize that the seminar is co-chaired, of course, as always, by Professor Christian Weber, whom I will pass the chair at the moment. But we also have a co-chair today, Professor Stefan Kaab from Munich. Welcome, Stefan, and thank you for joining us. Christian, please, if you could introduce the next speaker.
Thank you very much, Tom, and also thank you, Stan, for this very inspiring talk. And I think we have a very nice example of our format today here with one established figure in the field and one rising star. And with this, I introduce Svetlana Reilly. She's an associate professor at the Radcliffe Department of Medicine at the University of Oxford and also a BHF fellow in basic science. And so basically, she has also been working as a postdoctoral fellow in Barbara Casadei's lab, also, which is a well-known personality and leading personality in our European Society of Cardiology. And she has produced already in young years very seminal work on the molecular signaling in atrial fibrillation, in adverse myocardial remodeling and fibrosis in particular, standing out a role of microRNA 31 and also recently a role of calcitonin signaling. And with this, we look very much forward to your presentation. Again, I would like to reiterate, please post your questions in the chat function and we will come back to that in our joint discussion at the end.
Thank you so much for your kind introduction and for this great opportunity to speak today. I'm going to share slides now. Hopefully it works. Can you see my slides?
Yes, it works perfectly.
Mm-hmm. And the mouse as well, yes. Okay. Yes. So my talk is going to focus on our most recent studies that we published in 2020 on the role of cardiac calcitonin in atrial fibrillation. And I will hint towards possibly hormonal therapeutics in atrial fibrillation and what can be done. So the outline of my talk is shown on this slide. I will begin with a couple of slides of the background on atrial remodeling in atrial fibrillation. Then I will take you through some aspects of the discovery of cardiac calcitonin and will summarize with one slide our findings. So why is it so difficult to treat atrial fibrillation? Despite all the effort in the last decades, we are still kind of not succeeding with it. So perhaps the answer is in its progressive nature. So as we know, atrial fibrillation occurs or starts as short-lasting episodes, such as in post-operative and paroxysmal types of AF. And over time, it becomes persistent and permanent and irreversible. So how can we stop this vicious cycle and how can we target it? Obviously, progressive nature of atrial fibrillation is underlined by atrial remodeling that is associated with atrial fibrillation, electrical and structural changes in the atrium. Whereas electrical remodeling is very well described, and it's hallmarked by shortened action potential duration, as shown here, compared to the control patients. Structural remodeling is hallmarked by fibrosis. Fibrosis on its own is a major problem, and in a healthy heart of patients with sinus rhythm, we can maybe get up to 5% of fibrotic tissue in the atrium. However, when we quantify connective tissue component in patients with paroxysmal and chronic AF, we could see that the extent of fibrotic remodeling is increasing steadily. And the more fibrotic tissue we have in the atria, the more AF is maintained and it becomes more persistent. So my group especially is very interested in fibrosis, atrial fibrosis, as well as ventricular. And we pay a lot of attention to it because fibrotic remodeling on its own is a major problem. It's clinically irreversible to date, and over the last decade, the FDA has approved over 30 drugs for cardiovascular disease, yet there is not a single drug has been approved for targeting cardiac fibrosis. So there is no effective treatment to date. So in quest to understand the mechanisms causing atrial fibrosis and including fibrosis and atrial fibrillation, we went on to look at the literature and understood that actually complex hormonal abnormalities are often associated with atrial fibrillation and they are understudied. I wouldn't have time to go through each of them and how they contribute to atrial fibrillation. And I may refer you to a spotlight review that we've recently published in Cardiovascular Research. But I would like to focus just on one hormone, and it's calcitonin. So what is calcitonin? Calcitonin is a hormone of the thyroid gland that is produced into circulation, and its ultimate goal of calcitonin is to regulate calcium in the bloodstream, so to lower calcium level. How does it do it? It targets kidneys, bones, and intestine, and shuffle calcium either in or out to regulate blood calcium. In order for calcitonin to work, it has to bind to the receptor. Calcitonin receptor is a G protein coupled receptor class B. And upon activation of this receptor, receptor binds to GI, GS, or GQ proteins and causes a series of secondary messengers, activation or inhibition, and events in the cell. Interestingly, a few years ago, in Cell Reports, there was a paper published showing that knocking out calcitonin receptor in skeletal muscle, this skeletal muscle they used for the experiments, this event led to the increased fibrotic remodeling in skeletal muscle as shown here. It was exacerbated hugely. Although there was no data on myocardium or on the calcitonin receptor, on the role of calcitonin receptor in the heart, we were interested to look at it, especially considering that some GWAS studies suggest that polymorphism in calcitonin receptor gene is linked to the height and body mass, which are important risk factors for atrial fibrillation. So the hypothesis that we began to investigate at the time was whether calcitonin receptor may play a role in both atrial fibrosis and atrial fibrillation, but it can be possible only under one condition, if calcitonin receptor is actually expressed in the atrium. So we obtained human atrial biopsies from our cardiac surgery team and isolated atrial fibroblasts, because remember I mentioned that we are especially interested in structure remodeling accompanying atrial fibrillation. In this atrial fibroblasts, we confirmed that there is an evident expression of one isoform of human calcitonin receptor called 1A. And also we were able to detect that human atrial fibroblasts have a sufficient amount of protein by immunoblotting and immunofluorescence. And hence, if there is a receptor, so there may be an activation of this receptor and functional consequences of activation of this receptor on atrial myocardium and on atrial fibroblasts. So in order to investigate functional consequences of this calcitonin receptor cascade, we looked at extracellular matrix accumulation by fibroblasts, for example, collagen 1, and fibroblast proliferation and cell migration using wound healing assay. These experiments were done in vitro in human atrial fibroblasts. What we found that calcitonin appeared to potently inhibit a large number, I think it was 193 proteins in extracellular matrix accumulated by human atrial fibroblasts treated with calcitonin, as shown here by unbiased proteomics approach, which we performed in secretomes collected from these cells. And to confirm it, we also shown that collagen 1, which is in green here shown, is inhibited by about 46% in vitro in cells treated with calcitonin for 72 hours. So this data suggests that in vitro calcitonin via CTRs inhibits extracellular matrix accumulation. How about proliferation and cell migration? So real-time proliferation assay also showed that cells which were treated with calcitonin, which are in blue here, they proliferate a lot slower compared to the cells which were treated with vehicle or control. Similarly, cell migration was evidently slower in these cells, as shown here. After 24 hours of treatment with calcitonin, cardiac fibroblasts didn't heal the wound as fast as they've done in a control cohort of cells. Obviously, we were wondering whether all these effects at the protein levels may be underlined by transcriptomic changes. And we performed single-cell RNA sequencing of nearly 15,000 human atrial fibroblasts treated with calcitonin or with control vehicle. And as you could see evidently, so the control pink dots, so each dot is a cell, and CT-treated cells are blue. So there is no difference whatsoever. So we couldn't identify any, which was surprising. However, it suggests that perhaps calcitonin actions in human atrial fibroblasts are not dependent on the transcriptional changes and probably occur at the post-transcriptional level. So although all these in vitro experiments were encouraging and led us to think that perhaps calcitonin and calcitonin receptor signaling is important to modulate and control function of fibroblasts, we didn't know how it would perform in vivo. To explore it, we obtained global CTR knockout mice, and in Stanley's lab, we studied with mice, and we found that under basal conditions, calcitonin receptor knockout mice had a double amount of fibrosis compared to the control atria. And these profibrotic changes were associated with increased AF inducibility by about 26% in the mice which were lacking calcitonin receptor. So the signaling disruption of calcitonin receptor seems to be sufficient to cause atrial fibrosis and reduce atrial fibrillation. However, when we went back to clinical material to our human atrial fibroblasts obtained from patients with atrial fibrillation, we failed to find any beneficial effect of calcitonin on these cells. So we tried even supraphysiological concentration of calcitonin and saw no effect on collagen production. So we couldn't quite understand it. And the first step to understand why it's happening was to check calcitonin receptor gene expression, which was, however, unchanged in the presence of atrial fibrillation, suggesting that the lack of effect here is not due to the changes in gene expression of the receptor. So how about protein? Protein was modestly decreased by about 20%. However, it didn't make much difference, and it wouldn't be able to explain an absolute absence of the effect of calcitonin on human atrial fibroblasts obtained from patients with AF. So we were forced to perform localization studies using immunofluorescence. And surprisingly, we found that while control patients in sinus rhythm had a conventionally found or conventionally seen expression of calcitonin receptor at the surface of the cell, and some of it is inside the cell, like here. In patients with atrial fibrillation, receptor was everywhere. For example, here is in the nucleus with particular patient, but not on cell surface. So we can't even see staining there. And this is just one probably best example we have, but other examples show more distributed receptor localization. But one thing I think in common in atrial fibrillation is that fibroblasts from patients with AF do not have surface localized calcitonin receptor. This made us to conclude that in atrial fibrillation, disruption of the surface localization of this receptor causes or prevents a CT-mediated effect on profibrotic functions of fibroblasts. Another very important question which bothered me for many years was actually this one. We know that calcitonin is secreted by thyroid gland, and this is what is written in textbooks. However, a few clinical studies, in fact, probably more than a few, suggested and showed that, for example, this study shows that kids who were born with thyroid agenesis without thyroid gland or without thyroid, they have the same amount of calcitonin as kids who were born with thyroid. And the total calcium or ionized calcium is not altered in these kids. So this makes us think that maybe calcitonin is secreted elsewhere, and the thyroid gland is not the only organ that is primarily secreting calcitonin in human body. So obviously we were curious whether atria may express and secrete calcitonin. And so looking at the gene expression of calcitonin in the atrial myocardium, we evidently detected its presence. And isolating myocytes, fibroblasts, and epicardial fat from these biopsies also suggested that myocytes and fibroblasts express a calcitonin gene. The next step was to confirm whether myocytes or fibroblasts or both cell types may also produce protein and secrete calcitonin. So when we did ELISA for calcitonin, we found that ACFs, which are atrial cardiofibroblasts, they produce practically nothing, no calcitonin. However, atrial cardiomyocytes produce substantial amounts of calcitonin into media, conditioned media. Interestingly, to try to address this finding at the biological scale to see where calcitonin and how much calcitonin, how much myocytes produce calcitonin compared to other biologically meaningful systems like thyroid gland, we wanted to have a positive control, and obviously the best positive control would be thyroid cells obtained from a thyroid of healthy patient. However, we couldn't get that, so we got thyroid cancer cells, which were isolated from a female who died of a medullary thyroid cancer. And these cells are known to produce substantial amounts of calcitonin constitutively, so constantly. So when we measured calcitonin produced by these cells versus our atrial cardiomyocytes, we found that cardiomyocytes actually produce a lot more calcitonin than these cells over the same period of time. So this made us to think that perhaps atrial myocytes a major source of calcitonin in humans. So, and obviously it draws the intriguing possibility, which we obviously still have to confirm in subsequent studies, that atrial myocardium may represent a major extrathyroid source of calcitonin in human body. Going back to atrial fibrillation, we also were wondering whether cardiomyocytes produce maybe less or more calcitonin in this disease. And we found that actually in AF, cardiomyocytes produce around 6-fold lower amount of calcitonin compared to the control patients. And we wanted to test whether the impaired calcitonin secretion may be sufficient to cause spontaneous atrial fibrillation and atrial fibrosis in vivo in mice. For this purpose, we generated, in collaboration with Xander Verins from Baylor College of Medicine, atrial cardiomyocyte targeted knockdown or overexpression of calcitonin mouse. These mice were generated on the background of LKB1, which is liver kinase B1 deficient mice, which were available already. And what we found that when we knocked down calcitonin and with LKB1 knockdown mice, atria of these mice developed more fibrosis, as shown here on states of data. And when we did the rescue experiment, so we overexpressed 2-fold calcitonin in atrial cardiomyocytes of these mice. So fibrosis was back to normal. So it was rescued completely. Interestingly, when we looked at the spontaneous atrial fibrillation, incidence of spontaneous atrial fibrillation in these mice, we found that mice in red, which had lower levels of calcitonin secreted by atrial cardiomyocytes, had a doubled number of spontaneous AF episodes compared to the control LKB1 knockdown mice. And interestingly, overexpression of calcitonin in these mice prevented completely incidence of spontaneous atrial fibrillation in mice, suggesting that calcitonin receptor and calcitonin signaling is important to prevent atrial fibrosis and AF inducibility and susceptibility to spontaneous AF episodes. And both calcitonin and calcitonin receptor are equally important to maintain atrial function and structure in normal conditions. So to summarize, I would like to say that we identified a new and significant extrathyroid source of calcitonin in the human body. And we found that cardiac calcitonin controls atrial fibrosis and atrial fibrillation, as shown here on this diagram. We show that under normal conditions in sinus rhythm, calcitonin from atrial myocytes targets atrial fibroblasts, neighboring atrial fibroblasts, and by doing that, it controls atrial fibrogenesis, not allowing arrhythmogenesis. Whereas in atrial fibrillation, a low amount of calcitonin secreted by myocytes leads to the overstimulation of fibroblasts and higher production of extracellular matrix and increased atrial fibrotic remodeling, which begets atrial fibrillation. And we propose that maintaining physiological levels of cardiac calcitonin and perhaps of systemic calcitonin may benefit clinical management and treatment of patients with atrial fibrillation. This slide is probably the most important because I have to say that none of this could be possible without our collaborators. And we have a large number of collaborators. Especially, I would like to thank Lucia Moreira and Abhijit Takavali, who are the first joint authors on this work. And Abhijit works in Stanley Nattel's lab. And obviously, none of it would be possible without our colleagues from Baylor College of Medicine, led by Professor Xander Verins, who generated the LKB1 calcitonin knockdown and overexpressing mice. We wouldn't be able to do this study at the beginning without Professor Rachel Davey from Melbourne, who initially generated the global CT knockout mouse. None of it would be possible without Stanley's lab, and many people contributed to this work. And obviously, we were always relying on human atrial biopsies, which were generously carried out by our cardiac surgeons team. at the John Radcliffe Hospital at Oxford. And all proteomic studies were carried out by Professor Manuel Meyer's lab. And I would like to thank our founders, BHF, Oxford University, and Canadian Institute of Heart Research, and Canadian Foundation of Stroke. And I would like to thank our huge Oxford team as well, because all of them contributed to the study. Thank you. And thank you for your attention.
Well, thank you very much, Svetlana, for this excellent talk, and also thank you for sharing. So we have a open forum for discussion now. Maybe I can start, unless Tom or Stefan want to chip in. Maybe we come back to Stan's talk. So I think one important question that I had, and that also arose from some of the comments here in the chat, is basically the CANTOS trial, which features canakinumab antibody against IL-1 beta, which is sort of the main effector pathway of NLRP3. Obviously improves outcomes, but there's very little known as to how atrial fibrillation is affected in this. So can you basically comment or share some more in-depth knowledge about effects of canakinumab on atrial fibrillation?
Unfortunately, I can't. I don't know of any work that's been done on it, and I don't know of any data in the CANTOS trial. It certainly did not find a reduction in AF. I don't know whether it was actually examined or whether they even had enough cases to be able to know, or enough follow-up to be able to know. So in any case, I suspect that canakinumab would not be a great anti-AF candidate because of the risk of potentially serious side effects. But you raise a very important question, and I don't have the answer.
Yeah. There's obviously sorry, Tom.
Yeah, sorry to interject, but related to that, so Stan, where would you see the actual future targets in that pathway? Because you mentioned quite rightly canakinumab, and very upstream of the pathway is potentially problematic. But from your work, any other more direct targets and already possible candidates?
Yeah, so the resolvins are said to be relatively non-toxic, and some of them are in clinical trials. It definitely intuitively to me seems too good to be true that you could administer something that would be relatively non-toxic and simply promote resolution of inflammation. But at least theoretically, I wouldn't reject that possibility out of hand. Otherwise, we're still looking. So you would need to find a target that is specific enough to atrial cardiomyocyte inflammation or to fibroblast inflammation that it didn't produce serious negative effects on chronic administration.
I mean, you raised the possibility of side effects, but again, like if you would develop NLRP3 inhibitors, you would also have a likelihood that they have side effects in viral infection, for instance, or in also agonists are developed in cancer treatment. So basically, that there's probably
I'm not in any way discouraging any development. I'm simply saying that as a realistic target for atrial fibrillation, except for extremely unusual patients who have very difficult to control AF, even with the available non-pharmacologic measures, you wouldn't use something with a significant degree of toxicity. That's not to say that such products could be useful for other indications. Certainly they could. But for AF, we really need to be looking for things that are relatively innocuous. I would also point out, just as a matter of philosophy, that I don't think there's any one single miraculous bullet that's going to revolutionize all of the treatment of AF. And that from my point of view, in the long term, we're going to see progressive gains in AF management from a variety of directions and sources. And that too is why things need to be relatively non-toxic, because you can achieve things with relatively mild interventions that are also in general heart healthy, like losing weight, limiting alcohol intake, treating obstructive sleep apnea, getting regular exercise. And these are things which, although they seem fairly elementary, we're still not doing that well at them. So, of course, that's not the topic for discussion today, and we definitely need to continue looking for more specific targets. But nevertheless, there's a tremendous amount that can be done with the knowledge that we presently have that we're likely not doing so well yet.
Well, you also mentioned the resolvins, and maybe I can come back to that topic. I mean, there have been older studies also on fish oil consumption, the GIZI trial in heart failure, and I myself have basically started as an MD student in Peter Weber's lab, who was also with Alex Leaf, and so I just remember remotely that there may also have been a discussion on pro-arrhythmogenic effects of fish oil.
Yes.
How does it relate to these effects? And you also mentioned the fibroblasts, and are there also potential effects of resolvins and fish oil on other cell types, like and on calcium handling? That was also one question that arose here in the discussion.
Yes. So first of all, regarding effects in this model on calcium handling and drug effects on it, we haven't studied them yet. As you know, those are very large studies, and you can't do everything at once. So we have to be careful in what we choose to do. As far as the fish oil studies are concerned, globally, they've certainly been negative, and it's possible that that within the food, fish oils, of course, contain many compounds, and some could be beneficial and some could be harmful. Of course, the fact that that's a theoretical possibility doesn't prove in any way that it's a reality, but it certainly might explain some of the variable results and largely negative results that have been achieved. So again, I'm not naive enough to think that resolvins will necessarily be a miracle, but they certainly do have some interesting effects, and they're worth a study. That's what I would say. And there's certainly an interesting set of compounds that are worth looking into further.
Thank you. Stefan,
May I continue a little bit with the discussions? Thank you both, first of all, for your very inspiring presentations. And as time is marching on, I wanted to ask Svetlana on her findings on the aspect of the role of calcitonin and the heart as an endocrine organ, which I find fascinating. We've seen it in other aspects. Were you able to measure calcitonin level not only in the cells but also in the blood? Or could you envision differential levels of calcitonin by cardiac secreted calcitonin by measuring coronary sinus blood to be more specific? Have you done this, or would this be an idea?
All good questions, yeah. Thank you. They are all good questions, and actually, yeah, we've got some data unpublished on this, and yes, calcitonin is measurable in coronary sinus. And we are currently doing a clinical study with ablation cohort of patients in Cardiovascular, measuring coronary sinus, calcitonin, and other related biomarkers. So it's on the way, and hopefully one day we'll publish it soon.
Okay.
I'm not able to comment on the results yet because we are blinded. We're just waiting for patients to be recruited. So we're at the moment on 169 patients. So still a bit to go. But yeah, that's all undergoing. Yes, we also looked at the serum, just normal peripheral blood samples from patients undergoing cardiac surgery, because we know that postoperative AF is a major complication of cardiac surgery. And we do have very interesting data as well to share at some point with scientists and clinicians. Yeah, so I can kind of indicate that there is more going in with calcitonin than what we've just reported. So
yeah. But you mentioned, and I think that's true, that it's not about to have highly elevated calcitonin levels, but to maintain a homeostasis of the level, not to have too much fluctuations probably, and then to have specific locations like in the heart or in the, as compared to the whole body system, where you have kind of a compartment of different levels of calcitonin that then promotes either atrial fibrillation or the protection of atrial fibrillation. It will be interesting to see how this story evolves. That was actually one of the other questions, to look at ablation patients to take the blood, but it's great that you're already doing this, and I think this will lead the way to clinical applicability of understanding this. It's a very intriguing additional element. I think, as Stan said, it will not solve all the problems of atrial fibrillation, but it will add one little piece to the overall picture.
hopefully it will help.
There is a question. There is also a question. Sorry for interrupting on the chat on the time course of the calcitonin effect. So maybe just to save time on concerning both, you can address this as well. Sorry, I was.
Time course. Okay, so if it's related to our experimental time course, so it has been done under possible experimental conditions. So calcitonin was measured after 4-6 hours of cells incubated cellular buffer. We didn't measure any longer because obviously we didn't want to compromise cell viability. Cardiomyocytes are intrinsically difficult to keep in great shape. In terms of calcitonin time course, whether the question is related to postoperative versus paroxysmal versus persistent AF, I can say that all this study has been done on patients with persistent over 6 months of duration. All patients had long-standing AF. However, we are currently looking into postoperative AF closely, and we are getting some really interesting results. And we see that perhaps calcitonin signaling is not just relevant to the long-standing AF, but also to the short-lasting AF episodes. Whether it's a trigger when it's impaired or it's a consequence, I'm not ready to tell you yet.
Yeah, maybe following up also on this and also on Stefan's question. I mean, you show that it's myocyte produced and then the fibroblast is the target, so likely indicating that there's some local milieu for its effect. But then you have this beautiful tool of the atrial-specific knockout. So I was actually wondering, because calcitonin has been implicated also as a therapeutic tool and target in other diseases like diabetes and obesity. What about studying extra cardiac effects of atrial-specific calcitonin in your putting that in another disease model?
Yeah, that's a $1 million question, and that bothers me the most. Also, my peers like Stanley would say, "Don't waste your time." But I guess it's a very valid question. It takes some guts to prove it or disprove it. So we have generated an appropriate mouse right now in our lab to look at it, because actually we haven't proven that calcitonin secreted by atrial cardiomyocytes is actually secreted in bloodstream. Exactly. Also, this is a very attractive idea and obviously very intriguing if it is, but we have to prove it, and that's what we are currently doing.
Okay.
Certainly, if it does, as you suggested, Professor Weber, then the consequences of cardiac health for patients, for example, with osteoporosis or with Paget's disease or with diabetes, it would be massive. Therefore, probably it's more important to maintain cardiac health and thyroid. Forget about thyroid.
Yep.
Would be replaced. Sorry, probably not. But
yeah, we have some more questions here from the audience. Osteoporosis, you already mentioned. So basically, I think it's very hard and to follow up on all the questions, but I would also encourage the two of you maybe to also respond. And I think Sarah can be instrumental in that to respond to the questions later on by email, or if you get a chance here in the chat, because I think it's otherwise next to impossible to really address all these wonderful questions.
I would propose that as we have so many questions, because there is a lot of discussion this time on the question and answer, I would propose that that maybe Stan and Svetlana can stay a little bit longer and answer the questions, and Sarah, we will not close the session. We will leave it on until some written discussion occurs, because there is quite a lot of very interesting questions that I think are worth answering. I don't know if Stan and Svetlana are happy to do so, but I hope so. If not, I think the questions can be transferred by email. But we have a very vivid discussion today. Thank you.
Perhaps I can just start with a few of the questions that were addressed specifically to me. So Rosanna Bassani, I don't know if she's still there, asked about systemic inflammatory processes, whether they have a role in AF, given that they increase blood CRP levels. And there's definitely a strong association with a variety of inflammatory conditions in the occurrence of AF. So I think that's definitely a piece of evidence. Someone asked about ventricular myocytes and NLRP3. We haven't in our models, but Dr. Hillard Brown in San Diego has done extensive work, and actually turned out to be published at around the same time as Zander's studies and our studies. And there definitely seems to be an involvement of cardiomyocyte NLRP3 signaling in progression of heart failure and other cardiomyopathies. So this is probably a much broader theme than simply restricted to the atrium, although in patients with primarily atrial fibrillation, it seems to be the atrial cardiomyocytes that are primarily involved. And someone asked why, and I think that's an excellent question to which we don't really have an answer yet. Someone said,
what about resident macrophages also given their role?
Yes, so someone also asked about that, and whether inflammatory cells can have a role in the pathophysiology of AF, and they certainly can. Interestingly, in Zander's, in Nali's study, along with Zander, Warren's, they did create a macrophage overactivation model, which interestingly did not create a substrate for AF, for whatever that's worth. But I don't doubt that inflammation coming through inflammatory cells can potentially promote AF. I guess a clinical paradigm of primary inflammatory cell myocarditis would be some of the very severe acute myocarditides caused by inflammatory cell activation. And in those, atrial fibrillation is not a primary feature. That may be because the ventricles are so sick that we don't look at them. But in any case, it seems as though the atrial cardiomyocytes are a major component of the inflammatory system activation in AF. Let's see what else we have here. So Elizabetta Charbai asked about fibroblasts being involved in the protective action of the resolvins and the monocrotaline model. And we don't really have an answer because we didn't look at the types of cells that were being affected by the resolvin D1. So we don't know whether it's some sort of direct action on resolution of inflammation in fibroblasts, whether more likely, I would guess, it's cardiomyocytes that are having inflammatory signaling that's persistent and being suppressed by the resolvins that acts in a kind of paracrine way, in the opposite fashion from calcitonin, which seems to be produced by cardiomyocytes to protect fibroblasts. Perhaps the calcitonin acts as a brake against other substances that are produced by cardiomyocytes that activate fibroblasts. So that's certainly a very interesting area to pursue, but it's very speculative for the moment. Sharat Kumar asked about NLRP3 needed for viral infection clearance and inhibitors, and I definitely agree that inhibitors to NLRP3 action could have potentially very serious side effects. And indeed, with canakinumab, I think there is an increase. It's not large, but it's statistically significant and concerning, an increase in, I believe it's pneumonia and possibly even cardiogenic shock. So these things need to be used carefully. So I'm just looking, the best way to translate our results of NLRP3 to the clinical level. So I think the best way is to do more research to know what kinds of treatments might be effective and safe that manipulate the system. We're really only at the very beginning of this area. This has only very recently been shown. So what kinds of cells show NLRP3 activation? What kinds of treatments might suppress it without causing too many systemic effects? Those are critical questions before we can apply them clinically. I think I'll pass it on now to Svetlana. I don't know if she has other questions.
Thank you very much, Stanley. Yeah, plenty of questions. So one question from Ursula Ravens. I apologize, I misunderstood previously when it was read the question. So can you please comment on the time course of the calcitonin effects? So just to clarify to what I've said before, in vitro it's 72 hours maximum we could measure. However, in vivo, effects of calcitonin or time course of the effects would depend on whether the receptor is internalized or not. So obviously, if we try to stimulate the calcitonin receptor with administration of calcitonin IV or somehow else, so we should be aware of the fact that of the escape phenomenon of GPCRs, and calcitonin receptor is a typical GPCR which will escape eventually after 4 to 6 weeks of treatment with calcitonin. So this is just a biological process to preserve viability of the cells. So obviously the effect wouldn't last too long if we try to stimulate it like that. So we have to be a bit more clever and come up with something better than that.
The effect is controlling or affecting calcitonin.
Sorry?
Other factors controlling or affecting calcitonin production?
What controls production? Well, that's a difficult question, but we did have a look at a few potential pathways. One of them is actually microRNA 34A, 5P, because we know that this microRNA is associated with aging, and surprisingly, or not surprisingly perhaps, it came up as significantly upregulated in atrial cardiomyocytes in the presence of AF. And some in vitro assays suggest that it may target calcitonin. However, I don't believe that this is the only mechanism. Also, we know that calcitonin is regulated by estrogens and by hormones. So it's actually sex-dependent hormone to an extent. So males seems to have more calcitonin after the age of 65 compared to women. So there is something more fundamental to look at than just microRNA 31. And this is something we are looking at, yeah.
Wonderful. So I think we come to a close here of a really wonderful session and a really lively and stimulating discussion. Again, I would encourage Sarah maybe to pass on some of the questions that could not be answered by email. And I thank the two of you, Stanley and Svetlana, very much again. This was a wonderful occasion. There are well over 200 people in the audience, and this will also be live streamed in YouTube, and will continue to be available on YouTube for those who haven't managed to see it. And with this, also on behalf of Tom and Stefan, Tom, who heroically boarded a plane, during the last minutes of this of this session, I would like to thank you again for your participation, and stay tuned for the continuing program of this webinar between Cardiovascular Research and the Basic Science Council, which is still a huge success for us, and I think we're very happy to host this. Thank you.
Congratulations on a wonderful initiative. Thank you very much.
Thank you very much.
Have a
Thank
great
you.
day.
Thank you all. Bye-bye.